Hi, everyone. It's great to be back in San Diego after, I think, 3 or 4 years now, who knows. It's a real pleasure, honored to be part of this community. We've been thinking a lot at Notch Therapeutics about how we can maximize the benefits of T-cell therapies. I think we all know this community has delivered outstanding results, but as Jeff just spoke about, there are constraints to manufacturing and thinking about T-cell therapies that we have to address. And so I'm going to frame this presentation today to talk about how we're thinking about, how are you going to actually deliver this maximal benefit? The first thing you think about if you were to draw out a mind map is you would want unlimited T cells, an unlimited cell source, and there are two options for that. The first option is, you can extract cells from donors, and the other is that you use stem cells, either embryonic stem cells or induced pluripotent stem cells. And if you do the math, you quickly realize that although there's advantages to using donor-derived cells, stem cells truly offer that ability to achieve the unlimited stem cell source. There are caveats to that, though. You need to be able to deploy the advances that have been made in pluripotent stem cell biology, and you also need to somehow make the cell type of interest, in this case a T cell, without an embryonic fetal liver, without bone marrow, without a thymus, and do it all within a system that can be scaled and moved towards manufacturing. Of course, we all hear a lot about creating cells that are off the shelf. You want to make sure the body doesn't kill the cells that you're injecting, and you also want to make sure that the cells that you're injecting don't kill the body. So you need to do some engineering to address that. You want to think about the mechanism of action of the actual pathology that you're going after. So in that extent, you want to enhance the function of the cells. And you also want these things to be as safe as possible. And I think the enemy in cell therapies is the genome. So we have to monitor that genome very carefully, derive clonal and safe homogeneous lines. To do this, you can deploy gene editing. For those of you who have played with gene editing technologies, no matter how efficient and amazing and flashy your gene editing technology is, you need to be able to multiplex these things, or you're going to have development timelines that are exceptional. You need throughput in order to characterize and deliver on the promise of the gene editing technologies. And you need really robust analytics on the downside in order to be able to characterize the clones that you're generating. And to that extent, to tie it all together, you need to build systems biology approaches to understand what it is that you're doing to the genome of these cells that you're going to put into patients. And then finally, you think about simplifying the vein-to-vein manufacturing model. So the concept of taking T-cell therapies from being a process into a medicine. And to that extent, you have to think about quality from the start. Major investment goes into these products, so you need to be thinking about what the quality is going to look like from that starting material that's going to be the unlimited source. You need to keep it small footprint. You don't need to build out massive infrastructure to achieve this kind of manufacturing. And you want it to be centralized so that you can distribute over the world in frozen vials and deliver cells at the bedside. And to tie that all together, you want sophisticated engineering, top-notch engineering, if you will, in order to catalyze the differentiation processes into an actual product. And this sounds like wishful thinking, but this is exactly what our company is, Notch Therapeutics, and I'm excited to talk to you about how we're going to deliver on the promise of iPSC-based T-cell therapeutics today. So Notch is a spin-out from the University of Toronto. We were started in 2018, and in 2019 entered a pretty transformational partnership with Allogene Therapeutics to help deliver iPS-based allogeneic cells for their pipeline. We used that capital to then go on and build out the teams, bring on an executive team, and we raised an $85 million Series A in February last year. We are distributed over 3 sites based on sort of centers of excellence, headquartered in Vancouver, British Columbia, Toronto, Ontario, where we do our manufacturing, and Seattle, Washington, with our preclinical manufacturing. We are, and I'll talk to you about how the substance underneath this all, we are the world's expert in the control of Notch signaling during cell development. We are aiming to deliver T-cell function that is unprecedented from pluripotent stem cells. We do this all from the mindset of quality first, commercial manufacturing first, so that we can invest in these programs and have them actually yield products at the end. And we are deploying sophisticated gene editing technologies, analytics, and systems biology to design product profiles that will hopefully address the underlying mechanism of action of disease. And to that extent, Notch has emerged as an end-to-end iPSC therapeutics program or company that can enable multiple programs. So let's dive into it. Let's talk about where we start. If you're a pluripotent stem cell company, you really need to think very carefully about your starting material. To that extent, we've generated our own in-house, commercially compatible, GMP-compliant iPS cell lines that have been validated for lymphoid potential and that meet what we believe are some of the most stringent high-bar criteria for the release of iPS cells that you would use for commercial development. So in the left-hand panel there, you can see what these cells look like when we suspension adapt them so that they can be grown at scale to be the input material that you'll use into the differentiation process. And you can see that we're generating very high yields of hematopoietic progenitor cells from those cells, and that those are going to form the basis of the input material that becomes the T cell at the end of process. And we basically take these cells through stringent analytics, including the ability to form these hematopoietic progenitor cells and T cells at the end. So this brings us to the fun part, which is why this company is called Notch Therapeutics. We know from decades of literature that Notch signaling is an obligatory signaling pathway for the production of T cells in the thymus. And so what I'm showing here on the left panel, this is what we call in the company the Pikachu panel because it looks like Pikachu. So the Pikachu panel is basically showing from where that little arrow starts, is showing T cell development from the human thymic atlas. So this is RNA-seq data that's describing the emergence of T cells in the thymus. And so what our systems biology team did is they went and mined that data and said, hey, what's happening along Pikachu's body here when you look at Notch-responsive genes, knowing that, that Notch signaling is required for T cell development. And you can see that going from different cell type to different cell type, you have this differential expression of Notch gene regulatory networks, which teaches us that Notch signaling is critically important. And we also know that what else is happening in the thymus is positive and negative selection, so the signaling through that TCR is going to be critically important as well. And the sequencing of that is going to determine the outcomes of what is going to emerge from the thymus into your bloodstream. As I mentioned before, in this situation, we don't have the luxury of a thymus or a bloodstream or bone marrow where the cells derive. We're doing this all inside a dish. And so the core technology we licensed from Juan Carlos Zúñiga-Flucker's lab at the University of Toronto and Peter Zanstra's lab, also at the University of Toronto, was a technology that allowed us to provide the Notch signal to cells during development. This seems like an easy thing, but Notch signaling is not like a cytokine that you add to a dish. Notch is a biomechanical receptor that needs pulling and tugging and all sorts of other wonderful things so that it can be cleaved and sent into the cell nucleus where it does its job. And so, what we've done is we've basically gotten rid of the plate-based approaches that are currently being used to deliver that Notch signal. So on the left-hand side is an image of the OP9 feeder cell line that's being used by some of our competitors that expresses the DL4 ligand. That DL4 ligand is there, it's stuck, the cell makes its own decisions, and you have to basically, teach it. You have to train your cells against these cells that have a mind of their own, and from a manufacturing standpoint, if you're dealing with one cell already, dealing with two cells is a real pain. And you can see on the right, what we've done is we have used our technology to coat very simple beads. So we coat the beads with Notch ligands and an accessory molecule called VCAM1 that essentially modifies the effective dose of Notch signaling to the cells. The cells are rolling over the beads, they're happy as clams, and they start differentiating in a really meaningful way. What's important about this is we can liberate ourselves from the constraints of plate-based approaches, moving the cells into bioreactors, being able to control the timing, density of the Notch signals as the cell sees them through its stages of development. So the little mimic of whatever Pikachu is doing in this dish, we were able to control that signal. And we've been able to prove out the thesis that notch signaling matters in the last year of effort at the company. And these little colorful graphs here, basically, I want to actually just focus on the last one, which is if we look at the emergence of critical cell types during differentiation, whether it be the progenitor T cell that is going to reliably give rise to T cells, or the myeloid compartment, which is a cell that we don't want, by manipulating the amount of notch ligand and VCAM1 accessory molecule and essentially changing the effective doses of notch signaling, we're able to deliver the cell types we want and get rid of the cell types we don't want, which really helps manufacturing if you care about contaminants. Now, these cells that we take out further become T cells. These become alpha-beta T cells that can be stimulated with CD3, and we can time that signaling. We are going to be, generating a lot more data over the next 6 months to form a robust data set around this, and we're happy to share some of this data and the progress of the company under confidentiality. But suffice it to say, we've made a lot of progress in generating cells from iPSCs that can kill in a target-specific manner comparable to a primary T cell. So it's really an exciting time for Notch Therapeutics in its development of these products. And then we've also at the company, at our site in Toronto and Seattle, we have developed a very efficient platform for multiplex gene editing to enable multiple product iteration. And if you've ever tried to develop a gene-edited stem cell product, you know that one of the things that's going to hamper your timelines pretty impactfully is gene editing. And so we were able to multiplex our gene edits. We're able to generate enough clones and have a workflow that allows us to get the cells that we want at the end of process, run analytics through systems biology, and select the cells that we want at the end. And there's some smattering of data there that describes the high efficiency indels that we get from knockouts, and also fairly high efficiency knock-ins in iPS cells using a nuclease-based approach. And I think one of the things that we just have to hammer home, by moving this into our proprietary manufacturing platform, we're thinking small footprint manufacturing, where one vial of cells with a stable intermediate of a hematopoietic progenitor cell can then generate about 1 to the 12th cells that could be released as a product, which, if you think about it from a single vial, is about enough for 2,000 doses. So this is a really compelling value proposition, which we have actively demonstrated is possible in the company. So, we think that this could potentially be transformative for the field. We are under the management of a very experienced leadership team, and I wanted to just use this opportunity to highlight the people who have led the work. Emily Titus is our head of process development and technical operations in Toronto, an outstanding team leader and extremely knowledgeable. She's developed these cell lines that are just, coming from a pluripotent stem cell company background, it was the thing I was most paranoid about, and we've just knocked it out of the park. Christopher Bond, who was previously at Kite and Juno, leads our preclinical sciences program, and Deepika Rajesh, who recently joined us from Fuji Cellular Dynamics, is leading our differentiation efforts in Vancouver. And we have a very exciting team that somehow, deals with me on a daily basis. As we continue to grow this company, we're building out a pipeline of partnered and internal programs. This is a long haul. This is not, I think, as my first slides probably alluded to, this is not a small lift. So we're very open to discussions on partnerships. We can fully enable IPS-based programs, clinical grade from the get-go. So if you're interested in partnering, please reach out and let us know. We have a lot of experience managing alliances as justified through our Allogene agreement. So, just to close it up, we are world experts in notch signaling during T-cell differentiation. We're making T-cells from pluripotent stem cells, and it's a promising future for this field. So please, thank you very much for your time listening today, and please feel free to reach out to me if you have any questions. And it looks like I timed about 45 seconds for questions, so we can go from there. Thank you very much.